Hello, everyone, and welcome to today's webinar, Part 2, Utilizing the 6-Base Genome to Investigate the Dynamics of Enhancer DNA Methylation During Cellular Differentiation. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Biomodal. To learn more about our sponsor, please visit biomodal.com. Now, today's event, it's interactive, and we encourage you to participate by submitting any questions you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you may utilize this Q&A box, and we will assist you directly. I would now like to welcome our speaker, Dr. Emily Hodges, Department of Biochemistry, Vanderbilt Institute. For a complete biography on Dr. Hodges, please visit the Biography tab at the top of your screen. Emily, welcome. It's always such a pleasure to have you. You may now begin your presentation.
All right, thank you so much, Christy, for that introduction, and happy Halloween, everyone. For those of you who celebrate, I'm excited to be here to be able to tell you about our work and our longstanding interest in DNA methylation. So to start, I'm going to start with a very simple concept, and that is what we all know as the textbook understanding of DNA methylation and its role in gene regulation. So as we all know, DNA methylation occurs primarily at CG dinucleotides, so it affects cytosines in these CG dinucleotides. And the canonical understanding is that if you have methylation surrounding a gene, it's typically considered transcriptionally repressive, whereas if there's no DNA methylation at CG sites surrounding a gene, that's considered transcriptionally permissive. The problem with this model is that as we try to understand principles of gene regulation, we use these model organisms like yeast, worm, and fly, and they don't actually do DNA methylation. So being able to fit DNA methylation itself into models of gene regulation has been difficult. And also, there are plenty of exceptions that contradict the textbook understanding of DNA methylation. I'm going to be talking about those a lot today. The one thing that we do understand is that when mechanisms of DNA methylation are defective, so these are, for example, disruptions to enzymes that methylate DNA or demethylate DNA, it's disruptive to stem cell maintenance, differentiation, and genome stability. And so work from our lab and others have recently called into question or challenged what we understand is this canonical understanding of DNA methylation. So we typically measure DNA methylation genome-wide at single nucleotide resolution using what I would consider to be a gold standard method called bisulfite sequencing. And what that would produce is a track that looks like this. This is sort of a snapshot of the genome where every gold bar is a CG site, and the height of the bar represents how methylated it is. And then these horizontal bars, blue bars, represent regions that are hypomethylated. So you can see that most of the genome is highly methylated, where these gold bars are, and then that's interrupted by these scattered hypomethylated regions that we call HMRs. And if you compare these tracks across very diverse cell types, you can see that there are HMRs that are shared across very developmentally diverse cell types. We think that these HMRs, these hypomethylated regions, are established early on during development. There are other regions that are shared between a subset of cells. So these are lineage specific that are established as cells differentiate. And those that are more cell type specific, and we think those represent enhancer regions that are probably active in the cell type in which they are identified. So these HMR patterns really reveal enhancer patterns, and the specificity of these HMRs provide to us a record of development. So we've been really interested in how patterns of hypomethylation are established in enhancers. How are enhancers demethylated? And what is the relationship between DNA methylation dynamics and other aspects of the epigenome? And so that's something we've been recently exploring, and that I'll talk a lot about today. So here's this longstanding dogma. How does DNA methylation fit into models of gene regulation? So when cells undergo phenotypic transitions, genes are turned on and off. And this process involves gene regulatory elements like enhancers and promoters. So current models of gene regulation describe this as sort of a stepwise hierarchical process that involves sequential changes to the epigenome. And when genes are turned on, nucleosomes open up, transcription factors bind and recruit chromatin modifiers and DNA demethylases. DNA gets demethylated and genes are turned on. So this model really assumes that DNA methylation must be removed for transcription to occur. And the model also assumes that changes happen, all of these changes to these different layers of the epigenome are happening on the same time scale. So the work that I'm going to present to you today really started with this one observation that challenges this model. So we were investigating non-coding DNase hypersensitive sites, so regions that are open or representative of accessible chromatin in human embryonic stem cells. And what we noticed is that there's sort of this bimodal distribution of methylation that we observe in DNase hypersensitive sites. And so as we would expect in many DNase hypersensitive sites, you see that there are very low methylation levels. But there's also a lot of hypersensitive sites that were hypermethylated. And so what this would mean is that you have these potential putative enhancer elements that are at once both nucleosome free and open, but highly methylated. And so we kind of hypothesized, so one thing I want to point out is that when we look then at data from differentiated cells, these sites get resolved. So either the nucleosomes close or the DNA methylation eventually gets removed. So what we hypothesize is that these regions are likely undergoing some type of transition. They represent potentially bivalent, sort of like bivalent chromatin, where you have a sort of a poised or primed type of gene regulatory element. And the one thing we wanted to do is understand this better, but we really lack the tools to be able to observe how these regions transition from one state to another and be able to observe these two epigenetic states simultaneously. So in order to be able to do that, we developed a method that we call ATAC-STARR-seq, which essentially just combines ATAC-seq with bisulfite sequencing in order to be able to observe accessibility changes and methylation changes within the same DNA fragment population. And this was developed by my very first PhD student, Kelly Barnett. And we applied this method to a model of differentiation. So we started with a monocyte cell line that when you give it a drug called PMA, it differentiates to a macrophage within 24 hours. It's a great model because it's super homogeneous and it happens very rapidly. And so it allows us to take time points and perform ATAC-STARR-seq and then be able to measure chromatin accessibility and DNA methylation. And so I'm just showing you a subset of regions from this data set to make the point that within this time course, we observe really robust chromatin changes. We saw some regions that opened, and they begin to open within 30 minutes of treatment with the drug. But what was surprising is that methylation within these regions that underwent really robust accessibility changes, DNA methylation changes was very persistent, like it never changed. And this was really surprising to us. So the reason why it was surprising is because when we look at transcriptional responses in tandem with chromatin accessibility and DNA methylation, there were really robust transcriptional responses that tracked in lockstep with the chromatin accessibility changes, in spite of the fact there were no changes in DNA methylation. And this was true even when we extended the time course out to 72 hours. And the only time that we actually saw methylation go down in these regions that had been opened was when we supplemented the media with vitamin C, which is a really potent agonist for TET activity, which is the main demethylase active in this system. So I'm going to talk about that a lot more in detail later. So the one thing to note about these cells is that once they differentiate into macrophages, they are basically post-mitotic, so they no longer replicate. And so we thought maybe this is important. Maybe we need to observe methylation in cells that are undergoing active replication. And the other thing we were thinking is, well, in this sort of terminal differentiation state, maybe methylation changes are less important for the responses, the gene regulatory responses that are needed for these cells to function. So we decided to change to an earlier model of differentiation, and we chose to use embryonic stem cells. So we're now looking at embryonic stem cells, and we differentiated them into neural progenitors. And so this system is advantageous because you can do a really long time course. You can sample multiple time points over the course of 12 days. That's the sort of amount of time it takes for these embryonic stem cells to become stable neural progenitors. And the other advantage is that these cells continue to replicate during the 12-day time course. Another thing, when we look at single cell RNA sequencing data from different time points during the time course, we see that the cells differentiate fairly homogeneously, which means that if we observe epigenetic heterogeneity in our data, it's unlikely to come from cell population heterogeneity, which was an important thing that we needed to figure out before we started down this path. So we performed joint profiling of chromatin and DNA methylation with ATAC-Me over this 12-day time course, and we sampled 9 time points to begin with, going up to 12 days. And all of the work that I'm going to be presenting from now on was led by a senior graduate student in my lab, Lindsay Guerin, and her work is available. All of this stuff is available in her preprint that we posted on bioRxiv recently, if you want to check it out after the presentation. So the first thing we looked at was overall chromatin accessibility. So we determined the regions of change, so regions that were dynamically changing in terms of accessibility over the 12-day time course. And we just plotted this on this heat map. So in blue, you're seeing the regions that were highly accessible, and each column of the heat map represents a different time point throughout the time course. And so at the top of the heat map, you'll see delineated by this red line are the regions that are closing. And so they start out really highly accessible and they begin to close, and that transition sort of takes place between 48 and 72 hours. And on the bottom side of the heat map, those are the regions that begin to open and become very open by 12 days. And overall, this heat map represents about 38,000 regions that changed over the course of this time course. And the punchline from this is that most of these regions are actually enhancers or repressors. So many of them are outside of gene promoters. If we look at methylation in the same way, and we divide this heat map into the opening and closing regions, and we're looking at the methylation in this heat map are the white areas. The opening regions actually don't change at all. So even though they become completely closed by 12 days, those regions actually remain hypomethylated. Whereas, as one might expect, the regions that are opening undergo some type of demethylation process for the most part. So we can actually take this accessibility data and we can use unsupervised clustering to cluster the data into different groups according to their accessibility, temporal accessibility behavior. And so doing this, we came up with 7 groups of accessibility changes that track very well with the time points that we collected. And as I mentioned before, this data represents 38,000 regions that change over the time course. Most of these dynamic loci are intronic or distal intergenic, and they annotate to regions that are annotated as gene enhancers or repressors. Each time point has a unique group of accessibility regions, and those groups are enriched for transcription factor motifs that we would expect for these regions that are open early, like pluripotency factors like OCT4 and NNOG, transcription factor motifs that are associated with early neural progenitor specification, and those canonical transcription factors that are associated with the neural progenitor state, like PAC6. So, of course, our question was, if we define these different groups of accessibility changes, what is the temporal relationship between DNA methylation and these different groups? So represented here are the 6 different groups that I mentioned in the previous slide that we defined by unsupervised clustering. And the 2 lines that I'm plotting here are either the ATAC-STARR-seq data line, so accessibility in purple, or in green, methylation. And I will say, so that, because you're going to see a few of these plots throughout the presentation, standard difference is a way of standardizing the data so that we can plot it on the same scale. So we're looking at standard difference of the mean here. And we're looking at 6 of the different accessibility groups that we previously defined. And so what we can see is that there are these groups that begin to close early and are closed by the end of the time course. And as I mentioned before, that we saw in previous data sets, is that nothing's really happening to methylation in these regions that are closing. The other group of regions are regions that are transiently open. So we saw that regions open and close fairly transiently during the time course. And so we have those regions that open and close. And if you look at methylation in those regions, you do see that many of them undergo some type of demethylation during the time course. And then there are these late opening regions that also undergo demethylation in a concordant direction. And so we have these 3 different types of groups: opening, transient, and closing. And we can map those regions to a different methylation state, those that lose methylation, gain methylation, or are stably hypomethylated. And you can see the fraction or the proportion of regions that map to these different combinations here. I will say that it's about maybe 10% of our data of these regions actually gain methylation. So there's a small subset, but by and large, most regions are that are opening or changing in chromatin accessibility are losing methylation. So again, we have these different groups that either start inaccessible and methylated and become accessible and hypomethylated. That would be sort of a concordant transition. Regions that are accessible and hypomethylated and then become inaccessible and hypomethylated. Or these transient states where they're inaccessible and methylated starting out, becoming accessible and hypomethylated, and then losing accessibility but remaining hypomethylated. So this is an example of one of those groups of transient regions that opens and closes during the time course. And so this is the ATAC line, so the chromatin accessibility line. So you see that there's a really strong induction of chromatin accessibility that begins to happen as early as 24 hours. But in terms of methylation, the highest amount of demethylation is happening subsequent to the changes in chromatin accessibility, around between 48 and 108, which is equivalent to 4.5 days, hours of differentiation. And so DNA methylation changes are delayed. But we do see that they're at least in some cases concordant. So early on in the differentiation time course, when changes in accessibility, when accessibility goes up, DNA methylation goes down. It's just delayed relative to accessibility. And then we also observe discordant changes. So what I mean by discordant is in this example are these regions where accessibility goes down and DNA methylation seems to continue to decrease in spite of the fact that these regions are closing. So it's almost as if the hypomethylation of these regions is a record of past activity for these enhancers. And this is just showing this in a little bit more global detail. We have these different accessibility behaviors labeled on the x-axis of this proportion plot, and you can see that the proportion of these regions that either lose, gain, or have stable methylation. And so, and on the right, you can just see that how the changes in opening, these regions open over the time course, and they lose methylation. But the regions that are closing over the time course really don't gain very much methylation. So we really wanted to understand, what is driving these different patterns that we observe. And of course, transcription factors are really at the top of the hierarchy of gene regulation. So naturally, we looked at motif enrichment for transcription factors within these different accessibility groups. And as I mentioned before, there are transcription factors that are highly enriched in regions that are closing, and those that are very distinctly enriched in regions that are transiently open, and then those that are enriched in regions that open late in the time course. And to take this a step further, one of the advantages of having the ATAC part of the protocol in this data is that we can use the transposase cut sites as a way to estimate TF footprints. And then we can take those TF footprints and we can calculate methylation in those DNA fragments. So this allows us to more robustly identify regions that are occupied by transcription factors by being able to distinguish bound and unbound DNA fragments. And so this is just an example of a footprint for LHX2 where we are able to take the DNA fragments within these accessible sites and distinguish those that are bound and unbound by LHX2. And this is a larger representation of TF footprints over the whole time course, whole day 12-day time course. And each of the rows is the group of accessibility regions that we determined by clustering. And I'm showing you TF footprints for this POU power family of footprints, which contains several family members that are active at different time points during different periods of time during the time course. So that's depicted on the right, where you see the RNA-seq levels for 3 different family members. One that is OCT4, which is the pluripotency factor that goes down over the time course, and then another family member that's transiently expressed, and then the last one that is expressed very late in the time course, and the footprints of these family members over time. So if we look at all footprints and we ask, what does methylation look like in the regions that lose a footprint? And we ask, and we specify that we want to look only at regions that lose a TF binding event at 48 hours. What's happening to methylation of those sites over time? And for the regions that lose a footprint at 48 hours, very little happens. As I mentioned, very little methylation is gained in regions that were previously bound by a transcription factor. Whereas if we look at regions that gain a footprint at 48 hours, we see that they do indeed undergo demethylation, and but the methylation isn't really completely removed until after this TF binding event. So they undergo sort of this slow, what seems to be a slow demethylation process. And so The other thing I want to point out is that when we look at all of these different change dynamic behaviors between chromatin accessibility and DNA methylation, the one thing that sort of also stands out is that whereas with chromatin accessibility, you have so many changes that are happening very transiently and rapidly over this time course, but the difference with DNA methylation is that it appears to be mostly unidirectional and happening during a very specific window of time, no matter where you look. So the changes that we observe that seem to be with the highest magnitude of change are happening during a very distinct window of time. So just to summarize what I've showed you so far, here's where we were currently with our model, our new model of how DNA methylation mediates genome function. So we have our time course, we have our time points, and we're looking at changes in accessibility and DNA methylation over time. We observe that chromatin responds very quickly and continuously, and transcription of genes associated with these sites also is responding quickly and continuously over the time course. But DNA methylation changes are very distinct temporally from these changes. In fact, distinct DNA methylation changes are happening during a specific window of time, delayed relative to the chromatin changes. And another important observation is that previously active and accessible regions remain hypomethylated as a record of what happened to them in prior gene regulation events. So at this point, we're wondering, what are we actually missing? And obviously, we haven't actually looked at intermediate stages of DNA demethylation. And so if we looked at intermediate stages of demethylation, would this diagram look different? So if we looked at 5-hydroxymethylation, would this diagram look different? And so I will just explain really quickly that methylcytosine is removed by TET demethylases. So it undergoes a series of oxidation steps, and then combined with either replication or active removal through base excision repair, methylcytosine becomes unmethylated cytosine. So the first oxidation step leads 5-hydroxymethylcytosine to result in 5-methylcytosine to result in 5-hydroxymethylcytosine. And that's something that we're able to detect both globally and site-specifically. So the first thing we did is we did an ELISA measuring global hydroxymethylcytosine at these different time points to understand whether or not there was a change in hydroxymethylcytosine that's sort of coinciding with these DNA demethylation changes. And so what we observed is that there was a peak in 5-hydroxymethylcytosine that coincided with this window that I pointed out earlier, where we have the strongest level of demethylation happening. But it wasn't really telling us which sites were where DNA methylation was actively being removed by TET demethylases. The only way we would be able to observe that would be to have some type of sequencing approach that would allow us to distinguish 5-MC from 5-HMC. And so that's where 6-base genome sequencing comes in. So it's a orthogonal approach to whole genome bisulfite sequencing, except that there are different deamination steps and different molecular steps that allow us to be able to distinguish these two methylation states at single nucleotide resolution. So, so we were able to use this as an approach to in parallel to our ATAC-Me differentiation time course, to be able to understand where these sites of hydroxymethyl were occurring. And so what I'm showing you here are basically regions. So this is on the x-axis. These are like genome tracks, and this is the base chromosome location of these methylation tracks. And so on the top, we have 5-hydroxymethylcytosine proportion, and on the bottom, it's 5-methylcytosine. So the fraction of bases that are methylated or hydroxymethylated. And to zero in on one specific region, what we can see is that as you get, and each of these lines represents a different time point. So 0 is the dark line, 4 days is the purple line, and yellow is 8 days. We measured, we did 6-base sequencing at 3 different time points, one representing the earliest stage of differentiation, one representing sort of this intermediate transitional state, and one representing a state where the cells had already transitioned over to a neural progenitor state. And so what you can see is that as you get increases in hydroxymethylation, you get decreases in proportional decreases in methylcytosine. And the other thing to point out is that these scales are obviously very different. MC is really is very frequent, whereas hydroxymethylcytosine is very infrequent. So these scales are different, but the changes are very proportional to one another. So the first thing we did is for this 6-base data set is we asked what is happening to hydroxymethylcytosine in regions that we've defined by their accessibility behaviors. And overall, so these little thumbnail images are the accessibility behavior, so regions that are closing or are transiently open or opening late. And those on the right-hand lower box is looking at these static regions. And what's happening with hydroxymethylation is really interesting because it's tracking very closely with accessibility. In fact, it looks like it's mirroring accessibility in how it's changing, unlike methylcytosine that we saw in the ATAC-Me data. So if we look at this all together, it looks like in this one subset of regions, we have this increase in accessibility that happens towards the end of the time course. And methylation does change. You get demethylation in these regions. That's also happening. But hydroxymethylation appears to accumulate prior to increase in accessibility, resulting in the loss of 5-MC. So it looks like 5-HMC is starting early and lingers. So the whole time frame of demethylation is actually longer than we thought or we observed with the ATAC-Me data. And then if we look even within transcription factor footprints, we see something very similar. So this is an example of a footprint for this transcription factor that I've highlighted on the left. And we have the 3 different time points that we measured hydroxymethylation or performed 6-base sequencing. And what you can see is that we can distinguish with our footprinting method bound and unbound transcription factor binding sites and measure hydroxymethylation within those sites. At 4 days, you see an accumulation of 5-HMC happening that diminishes by 8 days and is highly depleted in the bound DNA fragments. So it looks like you get these increases in hydroxymethylation that accumulate before appreciable transcription factor binding, which is something that was very surprising and interesting to us. So this led to this sort of question. So the reason, I should say the reason why this was sort of surprising is that we always thought transcription factors lead the parade. So they bind and then initiate demethylation. But it seems like the initiation of demethylation might be happening either concomitant with transcription factor binding or potentially even before. So this led to a question: can we use 5-hydroxymethylation to forecast accessibility changes? And so in this last part of last few slides, I'll explain how we approach that. The first question we wanted to know is, can we use 6-base data to predict accessibility patterns at enhancers? And we did this in collaboration with some talented scientists at Biomodal who were also interested in similar questions. And so we asked, can we predict accessibility at promoters using methylation data obtained from the 6-base sequencing that we did? And of course, the methylation state and the accessibility state of promoter regions is very static compared to changes that are happening at enhancer regions. But it was really beautiful to see that we were actually successfully able to predict accessibility. So in these plots, I'm showing predicted accessibility versus observed accessibility at promoter regions. And the 3 different plots are just different types of training data that we used, either the total methylation with both methylcytosine and hydroxymethylcytosine, or with methylcytosine alone. And you can see that in all 3 plots, we observe a very positive correlation between predicted accessibility and observed accessibility. And you get some marginal increase in the efficiency of the model to predict accessibility if you use both methylcytosine and hydroxymethylcytosine. But we were really more interested in understanding whether or not we could use this data to predict changes in accessibility. And so, or accessibility in regions that are more dynamic. Because as I mentioned, promoters are way more stable in terms of methylation and accessibility. So I won't go into a lot of detail about this, only to say how we approached this in terms of our machine learning model design. As our input, we had, well, I showed you what the data was from promoters, but we also used our dynamic regions that we defined by their accessibility dynamics as input into the model. And we trained the model using data from all regions on all chromosomes, holding out chromosome 1 for our testing. So the models were trained on 90% of all regions and tested on the other 10% using chromosome 1 as our test set. And then we had 3 different time points. So we had 0, 4, and 8 days. And the training data that we used was either methylated cytosine, so the methylcytosine data from 6-base, the hydroxymethylcytosine data from 6-base, or a combination of the 2. And then we tested on different combinations of training time points and testing time points. So we had many combinations of testing and training data that we did, but I'm just going to show you some examples from one of them. So for this I'm about to show you, we did the training on 4 days and the testing on 4 days. And when we use methylcytosine alone, we are pretty good at predicting accessibility using methylcytosine alone. It's a positive correlation, but it's not nearly to the level of that we were able to do with promoters. If we use hydroxymethylcytosine, our model improves by quite a bit in terms of how we're able to predict accessibility. But if we use both methylcytosine and hydroxymethylcytosine, we get a greater improvement in the model in being able to predict. So what I'm going to show you on the next slide are focusing, instead of showing these scatter plots, I'm going to be showing you these Spearman correlations as a measure of model success. So then we asked the question, can we predict chromatin accessibility status at other time points using the methylation data? And it's really interesting because methylcytosine and hydroxymethylcytosine behave very differently in terms of these model predictions. So if we look at methylcytosine, we're really good at predicting accessibility in the past. So again, the training data is from 4 days, the testing data is from 4 days, either 0, 4, or 12 days. So if we train on 4 days for MC, we're really good at predicting accessibility at 0 days and less good at predicting accessibility at 4 and 12 days. If we use hydroxymethylcytosine, it's the opposite trend. We're actually really bad at predicting accessibility at 0 days, but better at predicting future accessibility states. And just as a reminder, we train this data at 4 days. And when we predict accessibility at 12 days, it performs even better than predicting at its own time point. And then, of course, if we use both methylcytosine and hydroxymethylcytosine in our training data set, those perform better than either of those states alone performed at predicting accessibility. They just contribute to the model differently depending on if you're predicting the past or the future. So what this machine learning allowed us to understand is that what we thought we were observing before is this sort of delayed changes in methylation actually can be informed by what we learned from being able to see hydroxymethylation. So we have these regions that are dynamically changing in terms of chromatin accessibility, and specifically looking at the regions that are opening. And if you just look at total methylation, which is what bisulfite sequencing does, it appears that demethylation is really delayed relative to chromatin accessibility. But with the added understanding of what's happening to hydroxymethylation proportionally compared to methylcytosine, it looks like the initiation of demethylation is happening actually pretty early. It's just that the completion of methylation is not happening until much later. So the total scale of change, or the temporal scale of change, is very different for methylation changes than accessibility changes. And so with that, I would just like to acknowledge my lab and the people that contributed to this work. As I mentioned, Lindsay Guerin, she was the lead on most of the work that I presented, and Tim Scott, a postdoc in the lab, did a lot of the computational work, and we again collaborated with many people from Biomodal who were very helpful in getting this off the ground. And yeah, I'm very happy. We have, please visit our website and our GitHub, and be sure to check out the bioRxiv preprint that we have. This paper is under revision right now, under review. So hopefully we'll get some good news soon. And I'm happy to take any questions. Thank you.
Emily, thank you so much for that excellent presentation. As you mentioned, it is now time to take questions. So to our audience, if you haven't already done so, please type your questions into the Ask a Question box, click Submit, and we will answer your questions, as many as we have time for. So Emily, we already have a few questions that have come in, so I'm just going to dive in. Starting with this one, in your opinion, what is the most exciting insight developed from the 6-base data in your study?
Yeah, I mean, I think it's just this the ability to be able to observe both hydroxymethylcytosine and methylcytosine sites specifically allowed us. I mean, there have been other methods that have allowed sequencing of high hydroxymethylcytosine. I think this one allowed us. It was a little bit more seamless to be able to get these libraries and do the sequencing, and so the quality of the data was really good. So I think that being able to observe something that we weren't previously able to observe provided a lot more insight into what's actually going on. And actually, it's not that we were necessarily wrong in our previous data. It's just that we didn't see the full picture. So yes, it is true that methylation changes are delayed relative to chromatin accessibility changes. And it is true that you might not require complete demethylation in order to get a robust transcriptional response. But it's not true that the initiation of demethylation isn't already happening. And so if you don't look at hydroxymethylation, you would never see that.
Thanks, Emily. Our next question. Overall, how has the 6-base data improved your understanding of the relationship between epigenetics and gene expression?
Yeah, it's that, because I think, we're only starting to appreciate that we need to consider these intermediate states. And of course, there are other intermediate states that we're not seeing here. We're not looking at the subsequent oxidation steps that get methylcytosine to unmethylated cytosine. So that includes formal and carboxyl intermediates that we're not observing here. But what this tells us is that we should be looking at these different intermediate steps and where you might see methylation. And we don't even know, so we don't even know very much about transcription factor binding to hydroxymethylation and the affinities that, there's been a lot of work that's looking at the impact of methylation on transcription factor binding sensitivity. So I think what this insight gained here is just saying, like, we need to be looking at both.
Thank you. Now, could you please comment on how 5-HMC and or 5-MC represent RNA expression?
Yeah, absolutely. Great question. So there have been some studies, not by our lab, but others that have looked at whether or not you can also use 5-HMC or 5-MC to predict gene expression changes. And I think accessibility, like we were able to show that we can predict accessibility changes using hydroxymethylation. And I think in methylation, it's also true that you can do this with gene expression.
Excellent. Very nice work. Do you think that DNA-me changes are more of a cause or consequence of transcriptional activity? And does it differ depending on the region, such as enhancers and promoters?
Yeah, that's a great question. And I think my answer 6 months ago would have been different if you asked me the same. Well, actually more than 6 months ago that we've had this data, but recently, in recent history, I might have answered the question differently. And this is the million-dollar question. Is DNA methylation instructive for transcriptional activity? And I would say that my initial hypothesis was that DNA methylation may not be instructive for gene transcriptional activity, but it is sort of a way to ensure cell fate stability or transcriptional stability. But now that we understand that the process of demethylation may be longer than we were appreciating before, and that initiation of demethylation, it's possible that initiation of demethylation is actually happening early and predictive of these changes. It's possible that in and of itself is a state that's important, instructive, potentially instructive for gene expression. So I think there's still a lot of things that we need to really understand. So the cause or consequence question, I think, is still there. But I think, now we know what we need to be looking at. And whether or not promoters and enhancers are different in this regard, we've, people have been looking at this for a long time and have seen that methylation, only a really small subset of developmentally specific promoters are getting are methylated and become demethylated. Most of them are constitutively, many promoters are constitutively hypomethylated because of their high CpG content. And so there's not a lot of hydroxymethylation at promoters for that reason. But so I do think they're different and they look different in terms of methylation changes. But again, I think we have a lot more to learn here.
Excellent. Emily, this question is 2 parts. I'm going to ask the first part first. Have you seen a significant shift in increase of HMC in promoter regions subsequent to changes in enhancer regions that affect those promoters?
So we haven't actually looked specifically at that question. I think it would be a good question to look at. A good thing to examine. I don't think that we've actually looked specifically at that. It's a great question. Yeah.
Okay. Well, the second part of this question is, are you seeing a cascade from enhancer region to promoter region?
Yeah, that's a good question. Instinctively, I would say that a lot of these promoter enhancer pairs and we don't actually, without having the chromatin contact data, we don't know which regions are actually forming these enhancer promoter pairs. But the ones that we predict are promoter targets of these enhancers, many of them are not really changing. They're already hypomethylated. They're just not necessarily transcriptionally active. So, but again, it's not something we've looked at, and I think it's something we certainly should look at.
Very good. Due to time, Emily, we're going to wrap on that final question. But do you have any final comments for our audience before we wrap?
No, I just think this is I guess the book's not closed yet, is what I would say. I think there's still a lot of things to understand, and I think what we've learned from our work is just that time is really understanding timescales of change is something that people should be considering. Yeah, thank you so much.
Absolutely. And Emily, I had this question come in a few different variations, so I do want to squeeze it in for our on-demand viewers. Does single nucleotide variants, SNVs, in enhancer or promoter regions, do they have any effect on MC or HMC profiling?
I would say certainly they can. But I know that it's not something that we ourselves have done. But this method allows you to distinguish variants and also be able to look at methylation in both states of methylation. So it's possible that you can see those things. Yeah, I don't know if it's more of a technical question or more of a biological question. So from the technical side, this would allow you to be able to look at both. From biological side, certainly, if you have variants in these regions that affect transcription factor binding sites or even specifically CpG sites, then certainly that can affect how you're looking at methylation. It can affect the methylation state of the region.
Sure. Emily, thank you so much. And thank you for taking time today to share your expertise in understanding how DNA methylation mediates enhancer function to drive cellular differentiation. I do want to thank our audience for coming today. Thanks for joining us and for your interesting questions. There were quite a few questions we were unable to answer. And additionally, those questions that come in during the on-demand period, they will be addressed by our speaker via the email address you provided at the time of registration. I want to thank LabRoots for hosting today, but mostly our sponsor, Biomodal, for underwriting today's educational webcast. Now, today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Now, when we close, this studio is going to redirect you to part 1. If you've missed part 1, we encourage you to register for that and view it. And additionally, please, if you have more questions, come back, watch on demand, and we'll get those questions answered. So to all our friends out there, thank you so much for joining us. We hope you have a beautiful day. Take care, everyone. Bye-bye.